Hello, students. Welcome to a new chapter Biotechnology and its applications. Biotechnology deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Three critical research areas of biotechnology are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme, creating optimal conditions through engineering for a catalyst to act, and downstream processing technologies to purify the protein or organic compound. Let's now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Let's look into the biotechnological applications in agriculture. The three options that can be thought for increasing food production are agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals like fertilizers and pesticides. However, for farmers in developing world, agrochemicals are often too expensive and further increases in yield with existing varieties are not possible using conventional breeding. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crop is a possible solution. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. GM plants have been useful in many ways. Genetic modification has made crops more tolerant to abiotic stresses, reduced reliance on chemical pesticides, helped to reduce post harvest losses, increased efficiency of mineral usage by plants, and enhanced nutritional value of food. In addition to this, genetic modification has been used to create tailor made plants to supply alternative resources to industries in the form of starches, fuels, and pharmaceuticals. Some of the applications of biotechnology in agriculture are the production of pest resistant plants, which could decrease the amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need of insecticides, in effect created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato, and soybean, etc. Let's see how is Bt cotton produced. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans like tobacco budworm and armyworm. Cleopterans like beetles and dipterans like flies and mosquitoes. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill the bacillus? Actually, the Bt toxin protein exists as inactive protoxins, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activated toxin binds to the surface of midgut epithelial cells and create pores that cause cell swelling and lysis and eventually cause death of the insect. Specific Bt cotton genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depends upon the crop and the targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene named CRY. There are a number of them. For example, the proteins encoded by the gene CRY1AC and CRY2AB control the cotton bollworms, and that of CRY1AB controls corn borers. Let's look into pest resistant plants. Several nematodes parasitize a wide variety of plants and animals, including human beings. A nematode, Melegnognine incognita, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents translation of mRNA. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements that replicate through an RNA intermediate. Using agrobacterium vectors, nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double stranded RNA that initiated RNA interference and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plants therefore got itself protected from the parasite. In this session, you have learned about the applications of biotechnology in the field of agriculture. With this, we come to the end of today's session. Thank you and have a nice day.